Welcome to our second lecture on photosynthesis. In this lecture, we'll introduce the light reactions of photosynthesis and focus on the biochemical activity of photosystem 2. So the light reactions occur in the thylakoid membrane within the chloroplast, and they strongly resemble the process that occurs within the electron transport chain of the mitochondria that we talked about earlier. So the light harvesting is going to start in photosystem 2. Unfortunately, photosystem 2 was the second photosystem that got discovered. Photosystem 1 was discovered first, and so it was named 1, but it actually comes in later in the process of the electron transport chain in the chloroplast. So we have to start at photosystem 2 and work our way across the electron transport chain inside the thylakoid membrane. We're going to harvest electrons. They're going to come from our water molecule, and oxygen will be produced in that process, plus the protons. The electrons are going to get harvested. Again, the light is going to come in, and it's going to be able to pop those electrons off. And they'll get transferred to a molecule of plastoquinone, which is very similar to the coenzyme Q that's involved in the electron transport chain of the mitochondria. And you can see it can exist in an oxidized or a reduced state. When it's reduced, it will drop off its electrons at the B6F complex. This one will also pump protons into the thylakoid lumen or thylakoid space, and the electrons will get transported to a peripheral protein that's called plastocyanin, and then they'll head over to photosystem 1. They get transported out to ferredoxin protein, and then a ferredoxin NADP reductase is going to transfer the electrons onto the molecule of NADP, making NADPH. So this is the final electron acceptor. Electrons are coming from the water, get donated onto the molecule of NADPH. The proton gradient that forms inside the thylakoid lumen is going to be utilized by an ATP synthase to generate molecules of ATP. So this is very, very similar to the electron transport chain process going on in the mitochondria. However, in the mitochondria, the electrons are being harvested off of our food molecules to be able to run the electron transport chain. In this system, the energy from the sunlight is being used to harvest the electrons, and it's pulling the electrons directly from pigments inside photosystem 2, and then those electrons get replaced as they're harvested from the water molecules. And so we'll take a closer look at this process. So photosystem 2 is also known as water plastoquinone oxidoreductase. So it's going to oxidize water molecules, it's going to pull the electrons off of them, and it's going to transfer them onto the molecule of plastoquinone. And so that's actually just shown up here in yellow, the quinone. You can see that photosystem 2, this is just showing half of it here. It's actually a dimer, so this is one half of the protein. You can see up in here it's got all of these green organic molecules that are trapped in here. These are chlorophyll molecules. You can see the carotenoids are shown in yellow. So this is kind of depicting their real color. And the carotenoids and the chlorophylls make up the antenna portion of the photosystem, and it's going to collect light, and the energy of that light is going to come down into the D1 and D2 sections, which hold a special pair of chlorophyll molecules. And so all of that energy gets funneled into this system until it's enough that it can pop off an electron from that special pair of chlorophyll molecules that's held in the D1, D2 subunits of this protein. And then they get transferred up through this molecule until they land on the quinone. And they're going to reduce the quinone. When the quinone is fully reduced, it can actually leave the molecule and it can carry those electrons to the next station. This manganese stabilizing protein down here is where you have the splitting of the water or the oxidation of the water, and it will replace the electrons that get lost in the D1, D2 pair. Okay, so this is another diagram that kind of shows a lot of those aspects of photosystem 2. So let's take a closer look at this photosystem. So if we pop this up here, we could see half of the protein in the last slide. So you can see that it is a dimer, and it's a homodimer, so it's the same subunits on each side of the molecule, and it's mirrored in this C2 axis right here. So the CP43 and CP47 regions of the photosystem, these are the antenna regions that contain all of those chlorophyll molecules that we saw a couple of slides back. And so this is where that energy is going to get gathered into. So the chlorophyll pigments, they have a long hydrocarbon tail on them. It's not shown drawn out here, but you can see it's a C20 that's hanging off of this end of the molecule here, and then the polyene structure located internally here. And it usually coordinates a magnesium ion at the center. And so sometimes the magnesium ion can be pulled out by a D-chelatase enzyme, and this forms a molecule that's now called thiophytin A instead of chlorophyll. It's essentially just a chlorophyll that's lost its metal. Okay, so you'll see reference to thiophytin in photosystem 2, and it plays a role in the electron transfer. And so the plastoquinone is the photosynthesis equivalent of coenzyme Q in the electron transport chain in the mitochondria. You can see plastoquinone also contains the quinone structure, and this is going to reduce to the aromatic ring structure, and both of these double bonds will become single bonds, and you'll have the alcohol groups out here to form the plastoquinol in the fully reduced state. So this molecule then is the final electron acceptor from photosystem 2, and it will transport the electrons to the next station in the electron transport chain. So now if we go back to photosystem 2, we can take a deeper look at this molecule. So we said the antenna sections are the CP43 and 47 regions that have all of those chlorophyll and other pigment molecules that are harvesting light. So they direct that light energy towards the reaction center, which is housed in the D1 and D2 subunit as they're coming down in here. So this reaction center contains two chlorophyll molecules that are known as the P680, or the special pair. They're called P680 because that's where they absorb the light the best in the wavelength spectrum, is 680 nanometers. So when the P680 reaction center chlorophylls receive the energy from a photon of light, it's enough to actually free up an electron from that molecule. And this electron then gets transferred to plastoquinone. Once the plastoquinone accepts two electrons and then two protons also from the stromal side of the chloroplast, it will be fully reduced. And when it's fully reduced, then it can leave from the photosystem 2 and carry those electrons to the B6F complex. The electrons, though, that get bumped off of that special pair of chlorophyll molecules at the D1, D2 center, they have to be replaced from the electrons from water. So the water has to ultimately be oxidized to keep this process running. Otherwise, you would lose those electrons from the chlorophyll molecules, and then you couldn't do it again, right? And the system would be over. So the small subunits down here, the PSBO, U, and B, this is the region that stabilizes the manganese core structure that's involved in the oxidation of water. And you'll note that that's coming from the lumen side of the thylakoid, and then the cytoplasm or the stromal side which is actually the inside of the chloroplast, would be on this side. So if we only color the pigment molecules in here, we can see the movement of the electrons a little bit better. And so what happens is the photons of light are coming in here. You see our little lightning bolt, and they get funneled into the D1, D2. And this is our special pair of chlorophyll molecules. And an electron is going to pop off of here when there's enough energy. It passes through a molecule of pheophytin. And as it does that, it ends up on the quinone. And the first plastoquinone is called plastoquinone A. And this one is permanently bound into the structure of the PS2. It cannot leave. It is a prosthetic group, so it's very tightly bound. And this second one, this plastoquinone B, this is the one that is a mobile cofactor. So once it accepts the electrons from quinone A, it can actually leave the photosystem too. So it has to kind of go through two quinones to be fully reduced and then leave photosystem 2. So once the electrons have popped off here, we have to actually pull them off of a water molecule to be able to replace them from the special pair that has lost the 2 electrons from here. They got transferred up and donated to reduce the molecule of plastoquinone. So those electrons are coming from water, and they're coordinated by a manganese cluster. And so the manganese cluster is shown in a little bit more detail over here on the right. And so you can see that there are a number of manganese atoms that are associated in here, as well as oxygen atoms and a calcium metal ion as well. And these help to coordinate the water molecules, and we'll see that in a little bit more detail on the next slide. So this depicts the activity of the manganese center to oxidize the water molecule. So first, 2 water molecules are going to come into this system, and they're going to coordinate with the manganese center. And so you can see they coordinate with one manganese out here, and then with the calcium metal as well in this square structure. Then the first electron is going to be harvested. That is going to be coordinated by this manganese. So you can see the manganese can support that oxidation step and goes to an oxidation state of +3 from the +2. So you're losing that electron, and a proton is also coming off at this point. And so the manganese can help stabilize the hydroxyl group that remains on there. At the second step, you lose another electron from the system, and you can see that's coming internally off of the manganese core structure. So another manganese is jumping up an oxidation state, showing that it's lost an electron. So that's that magnesium going to a 4+ state. And at this stage, another electron and another proton, my proton's missing from that step, but another proton will also exit here. And you can see that our manganese is jumping again from a +3 to a +4 state. We've lost that other proton, and you can see that we formed a double bond with the oxygen molecule from the first water. Then we continue to lose electrons here. This one again coming from the manganese, and it's going to enable the nucleophilic attack of our oxygen molecule on this other oxygen, because now we're at a very oxidized state for our manganese, and it's going to be drawing or pulling those electrons away from the oxygen. That doesn't happen very often, but in this case it's going to happen, and it's going to allow this oxygen molecule to mediate attack onto that oxygen molecule and form this bond. When it does that, the electrons from the double bond here can push down to the manganese and replace the ones that were lost during our manganese process. So we gain a couple of back as this one attacks and joins on this place here, and we lose that proton off of our water molecule. So the electrons from here end up popping over and forming that bond here, and we gain back the electrons to the manganese. So at this point, the oxygen can pop off, and this proton can pop off here, so this bond is going to fall in here and make the double bond with the molecular oxygen, and the proton will pop off. And then we're reestablished. The electrons will flow down here and recreate our +2 state and our +3 state here. So we've got 2 electrons that get distributed back into the manganese central core structure. So metals are great because they can accept single electrons at a time and change their oxidation states, especially something like manganese, which is a transition metal and has multiple oxidation states that it can take. So in summary then, the light energy is going to be absorbed by photosystem 2 in the antenna region. This is a very simplified diagram here. And the energy is going to go into that special core. It's going to allow the electron to pop off of the special chlorophyll molecules, gets transported to a molecule of quinone, which is our primary electron acceptor in photosystem 2. And then this can leave the molecule and transport those electrons to the next station. The electrons have to be harvested back from our water molecule so that they can be donated back to the special pair of chlorophyll molecules in the reaction core. And byproduct of this, thankfully, is oxygen, and that's the whole reason we exist, is because algae and plants evolved to do this process and produce the oxygen that we can now breathe. Notice, too, that it's also generating a couple of protons. The protons, again, are very important for being able to generate a proton gradient that builds up in the thylakoid space. This gradient is going to be very important, again, for the ATP synthase to run and be able to produce ATP. And we'll get to that in our next lecture section.